Almost everyone who sees a placenta is more interested in the baby it's attached to. The placenta, if you notice it, is a rather plain and puddingy organ. But if you dissect away the pale grey maternal tissue that covers the placenta, you'll see that it's actually one of the most beautiful organs in the body, because it's made up of thousands upon thousands of feathery fingers which float in the maternal blood. We call these fingers villi, and they're the life support for the growing baby, because they allow nutrients and oxygen to cross from the maternal blood into the placenta and then through the umbilical cord and into the baby. How well this happens affects the health of the baby during pregnancy, after birth, and into adulthood, and it's mediated by cells of the immune system. So let's meet some of those cells now. We can, broadly speaking, divide immune cells into two camps, innate and adaptive. Innate immune cells are very ancient, and we find them in organisms as simple as sponges. They recognize molecules that are shared by entire groups of pathogens, and they rapidly respond by causing inflammation. But they respond the same way every time. Adaptive immune cells are a more recent innovation in evolutionary terms. We first see them appearing about 500 million years ago in sharks. Adaptive immune cells are able to recognize any molecule that can possibly exist, and they can remember it, so that the second time they see it, they respond more quickly. It's this phenomenon that we're taking advantage of when we make vaccines. There are two main kinds of adaptive immune cells, B cells and T cells. B cells make antibodies, which are large molecules that bind to pathogens and toxins and prevent them from doing us harm. T cells look out for pathogens inside other cells of the body, and they do this with the help of a molecule called the MHC, which acts as a kind of window on the cell. The MHC grabs a sample of everything that's being made inside the cell and carries it up to the cell surface where the T cell can see it. The T cell will then react to anything that's not usually being made in the body. The word that immunologists would use for that is non-self. So, for example, if a cell is infected with a virus, the MHC will grab some viral molecules and show them to T cells. The T cells will recognize those as non-self and attack the infected cell. But here's the thing. T cells also recognize other people's MHC as non-self and will attack that. And that's the reason that organ transplants are usually rejected by the immune system. Back to the placenta, and you can see how this might cause a problem, because the placenta belongs to the baby, who is, of course, genetically distinct from the mother. And if, right now, I needed a kidney transplant and my son volunteered to be a donor for me, my T cells would recognize his kidney as non-self and attack it. And yet, he lived for 9 months inside my body, and my immune system didn't attack him then. So how does this occur? This is a question that was first considered by the immunologist who first started thinking about organ transplantation, Petter Brodin, way back in 1953. And even with so long, we still don't have all the answers. But Brodin started us off by saying, well, during pregnancy, maternal immune cells are never in contact with the baby itself, only ever with the placenta. So perhaps the placenta can act as a sort of immunologically neutral barrier that the baby hides behind. And indeed, it turns out that villus cells of the placenta, unlike almost any other cell in the body, just don't express MHC. So they're invisible to the T cells that surround them in this pool of maternal blood. Now, I've talked quite a lot about the pool of maternal blood, and some of you might be wondering where it comes from. Surely I don't have a pool of blood just sloshing about in my uterus the whole time. And no, you don't. Outside of pregnancy, blood flow to the uterus is very similar to any other organ in the body. In through arteries, through capillaries where oxygen is released into the tissue, and out again through veins. But during pregnancy, placental cells called extravillous trophoblast cells invade into the lining of the uterus to form this pool of blood. I've drawn extravillous trophoblast cells here in orange, migrating away from the tips of the placental villi, which are in yellow. And the extravillous trophoblast cells invade into the arteries from the top, opening them out, but they also invade through the tissue itself and surround the arteries, causing their muscular walls to dissolve away. So by about 10 weeks of pregnancy, we have this situation where the cells of the arteries have been entirely replaced by these placental extravillous trophoblast cells, and the arteries themselves are these wide, low-pressure vessels, and at their ends, this pool of blood in which the placental villi sit. I've mentioned before how villus cells of the placenta don't express MHC, so that makes them immunologically invisible. So being invisible is clearly an option if you're a placental cell. And yet, extravillus trophoblast cells don't do that. Instead, they express a special and unique combination of MHC molecules that isn't really seen by T cells, but is specially calibrated to allow them to communicate with innate immune cells in the lining of the uterus, NK cells and macrophages. And how well this interaction occurs affects how well the placenta implants. This is important because if it doesn't implant very well, these vessels don't open out and don't form a nice pool of blood, and that means the baby doesn't get as much nutrients and oxygen as it needs and may end up being smaller than we would want it to be. And that's important not just in pregnancy and at birth, but also into adulthood, because adults who were small babies are more at risk of cardiovascular and metabolic diseases, and they tend to die younger. So the blueprint for your future health was laid down by this interaction that happened between your placental cells and your mother's immune cells that began probably before she even knew she was pregnant. From the second trimester onwards, the placenta supports the future health of the baby in another way, which is by allowing antibodies to cross from the mother's blood into the baby's. Now, antibodies are large molecules, and large molecules don't usually cross the placenta. But antibodies can, because over the course of 55 million years, primates like us have evolved a special shuttle in the placenta that allows it to happen. And the shuttle works like this. Villus cells in the placenta take up components of the maternal blood in little bubbles called vesicles. Inside these little bubbles, there's a molecule called the neonatal antibody receptor, which binds to maternal antibody and ensures that it's transported across to the baby's side of the placenta, where it's released and eventually makes its way into the baby's blood. This means that when the baby's born, they have a selection of antibodies that were made by their mother against threats that she experienced during and shortly before her pregnancy. This is useful because newborns are very vulnerable to infectious disease, and this transfer of antibodies means that the mother can lend her baby some protection to protect it during this time until it's able to make its own immune response. But we can also take advantage of it. So any of you who've had a baby since 2012 will have been offered the whooping cough vaccine during your pregnancy. The logic behind this isn't that whooping cough is more dangerous in pregnancy, but rather in 2012 we had a big whooping cough outbreak in newborns, and we wanted to find a way to protect them somehow until they were old enough at 8 weeks to get their own vaccines. So the plan was to offer whooping cough boosters in pregnancy to boost maternal antibody so that more antibody could cross the placenta into babies and protect them after they were born. And it worked. After the vaccine was introduced, whooping cough cases in newborns fell by 80%. The other two vaccines that we offer in pregnancy, those we do actually offer because of flu and COVID, and those we do actually offer because flu and COVID are more dangerous if you catch them when you're pregnant. But when we look at the health of babies, it's a 2-for-1, because it turns out that babies born following COVID or flu vaccination in pregnancy are themselves less likely to catch and get seriously ill with COVID and flu. So we've talked a bit about how maternal antibodies can cross the placenta and protect babies against infection after they're born. But why do babies need this protection? Well, the pat answer is that babies are underdeveloped in lots of ways. They can't walk, they can't talk. Is it really so surprising if they can't raise a proper immune response? But it might not be as simple as that, because perhaps a newborn's immune response is perfectly calibrated to the challenges that a newborn faces. And one of the big ones is that the baby goes from being sterile inside the uterus to coming out and suddenly being colonized with loads of bacteria, which scientists call the commensal microbiota, but you might also have heard referred to as friendly bacteria. Now, an adult who was suddenly colonized with loads of bacteria, friendly or not, Their innate immune cells would raise a strong inflammatory response, which would kill the bacteria, but would also cause extensive tissue damage. Well, babies don't want to kill their new friendly bacteria, and they certainly don't want to experience extensive tissue damage. So perhaps it makes sense that the baby's immune response is a little bit less strong than an adult's in this respect. So the bacteria colonize the baby, so the baby allows the bacteria to colonize it, but the bacteria aren't passive players in this process. We think that they're talking to the immune system too. And one of the ways that we think this happens is that they can dampen down aspects of the immune response that can cause asthma and allergies. The reason we think this is the case is because babies who have lots of courses of antibiotics when they're young tend to have a less diverse community of bacteria in their gut, and that correlates with them being more likely to have an asthma diagnosis before they're 5. I'm not saying, of course, that we shouldn't treat babies with antibiotics if they need them, but rather we should perhaps think a little bit more about how we can put these friendly bacteria back in afterwards. So I've mentioned 3 ways in which the immune system is important in pregnancy and early life. Immune cells mediate the implantation of the placenta, which has knock-on effects on the health of babies during pregnancy and all the way through into adulthood. Antibodies crossing the placenta can protect babies against infectious diseases after they're born. And friendly bacteria colonizing us can influence our immune responses, including into later life. Of course, there's lots more to discuss, so let's now hand over to you, our audience, and to the panel to talk about it some more.
And we also have Professor Lindsay Hall. Tell us about your job.
Hey everybody, I'm Chair of Microbiome Research, which Vicky's done a fantastic job already introducing. So I'm a microbiologist, and we're really interested in how maternal gut bacteria influence fetal development, and also how bacteria, these friendly bacteria, are really important for maintaining health during early life as well.
Thank you. And we have Dr. Beth Holder.
Hi, so I'm Beth Holder. I'm a senior lecturer in maternal and fetal health at Imperial College London. And my research is focused on studying the communication between the immune system and the placenta in pregnancy. This involved both studying extracellular vesicles, which are little bubbles released from cells into the extracellular space, and also studying how antibodies cross the placenta to help protect the newborn.
Well, thank you for coming today. And we have Professor Chrissie Jones.
Hello everyone, so I'm from the University of Southampton. I'm a children's doctor and I specialise in infections in children and immunology. And I'm also a scientist, and I run clinical trials and study the best ways that we can protect women against infections in pregnancy and babies in early life. And together with Beth, we also run a network called Imprints, which is a global network of people who work in maternal vaccination.
Thank you. And finally, we have Professor Petter Brodin.
Yes, hi everybody. I'm a professor at Imperial College and a children's doctor as well, specializing in the immune system of young children, and particularly how it's regulated and shaped by the environmental exposures that we've just heard about early in life.
Well, what we're going to do first of all is to look at pregnancy, and then later on we're going to look at what happens after the baby's been born. But thinking of pregnancy first of all, so Vicky, from what you're saying there, is the mother's immune system actually weaker during pregnancy than usual?
That's been a popular idea, but it's actually a little bit more complicated than that. So I did say in my talk earlier that you're more at risk of getting very sick with COVID or flu if you catch them during pregnancy. That could suggest, a certain level of suppression of the immune system. But we also have to remember that the heart and lungs are under a great deal of strain in pregnancy, so that probably also contributes. And there are lots of infectious diseases that we are not more vulnerable to during pregnancy. If we look at the flip side, autoimmune disease, so that's when your immune system is, we might say, stronger than it should be. If we think the immune system is getting weaker in pregnancy, then autoimmune diseases should get less bad. And that is true for some autoimmune diseases. Multiple sclerosis is a good example. But some autoimmune diseases actually get worse during pregnancy. So lupus is an example of that. And then finally, if we think about vaccination in pregnancy, broadly speaking, we respond just as well to vaccines when we're pregnant as we do when we're not pregnant. So from all of that, I think we can probably say that it's not fair to say that the immune system is generally weaker in pregnancy. It is different in some ways, and some of those ways do leave us more vulnerable to some infections, which is why it's important to take care and to get vaccinated against things that you can protect yourself against. But I would say it's more accurate to say that the immune system changes in pregnancy rather than that it's weaker.
Right, so different rather than weaker itself. And Beth, one of the things you often hear about in pregnancy is the importance of the rhesus blood type. Can you explain what that's about, why that matters so much, why that's related to this?
Yeah. So when we hear about blood type, you may hear people say that they're AB positive or O negative. So the letters refer to their blood type, and then the positive or negative refers to their rhesus status. So somebody who is rhesus positive has the rhesus antigen on the surface of the red blood cells, and somebody who's rhesus negative is lacking that antigen. So similar to Vicky's story of the organ transplantation, if you took somebody who was rhesus negative and gave them blood of somebody who's rhesus positive, their immune system would respond to that foreign antigen and recognize it as non-self, and would start producing antibodies against that antigen and destroy the red blood cells. So as Vicky also introduced, the placenta in pregnancy separates the maternal blood and the fetal blood. But there are some cases when there can be transfer of red blood cells from the fetal circulation into the maternal circulation. So late miscarriage, abdominal trauma, most commonly during delivery, there can be transfer of small amounts of blood from the fetal blood into the maternal circulation. And when that happens, the mother can produce antibodies against that antigen and destroy those red blood cells. So where this becomes important is when the mother is rhesus negative and the baby is rhesus positive. And in that current pregnancy, it's not normally a problem because those red blood cells that transfer across, say during delivery, will be destroyed and the baby will be born safely. But if that mother then has a subsequent pregnancy, her body will respond and produce those antibodies that will cross the placenta, as Vicky introduced, and will start destroying the fetal red blood cells. And this results in anemia of the baby and also very severe jaundice, which can result in severe brain damage and other complications. So what we do is for all pregnant women, their blood is tested in the first trimester to identify their rhesus status. And if someone is rhesus negative, as a precaution, they're all given an injection of anti-D antibodies at 28 weeks. And what those antibodies do is they will attack any red blood cells that cross into her bloodstream very quickly before her immune system has a chance to recognize those red blood cells and respond. And this means if she has a subsequent pregnancy, again with a rhesus positive baby, that baby won't have any complications.
So this must have made a real difference once this was worked out, because I'm assuming we didn't always know this.
Correct. And I wish I knew when we identified that, but I don't know.
Fair enough. Petter, is it fair to say the fetus has its own immune system?
Oh, most certainly, yes. And viruses, for example, can transmit across, and the baby's immune system can respond in the fetus. I mean, the fetus can respond to those viruses. It does vary, though, across the life course of the fetal life. So in the very, very beginning, for example, the adaptive immune system is not really detectable, but the innate cells are there very, very early. And then as the fetus grows, this development happens successively over time.
And what kind of immunity would the mother be transferring to the
the transfer
fetus?
Is the antibodies that Vicky introduced nicely. Mm-hmm. So there's no other transfer really. There are a few cells that can pass over, but I think that becomes a bit complex to get into here. So the transfer is primarily the antibodies, but the fetus itself is equipped with an immune system that can respond to viruses, for example.
And Vicky, is this something that can go wrong sometimes?
Oh, there are lots of things that can go wrong.
Yeah.
I suppose the most important thing to say is, during pregnancy, the mother and baby, they're a single unit. So anything that's going wrong for the mother is also going wrong for the baby. So infections, or if you say have an autoimmune disease that's not well controlled, all of those things are bad for the baby. And so we should always remember to take care of our own health as mothers when we're expecting babies, because we're worth it, but also for the baby. But kind of thinking now about things that are a little bit more harder to intervene on, one of the things that I'm really interested in is this idea that the immune cells in the lining of the uterus help the placenta to implant. And I'm particularly interested in one of the innate immune cells, which is NK cells. And NK stands for natural killer. In the blood, they are very good at killing virally infected cells. In the uterus, it's actually a bit of a misnomer, because in the uterus, even though they look like the blood cells, they're really bad at killing, even at killing cancerous cells, and they certainly don't ever kill cells of the placenta. But what we think they do is they kind of give signals to the extravillous trophoblast cells of the placenta to help them move into the lining of the uterus, opening up those vessels to supply the placenta with blood. And I mentioned that this sometimes doesn't happen properly, and we can get a baby that's smaller than we might expect. We might also get a disease called preeclampsia, where the placenta, because it's being starved, sort of releases distress signals into the blood. And we can also, if it happens very early, get an early failure of pregnancy, which would be a miscarriage. And one of the pieces of evidence that these cells are really important is because certain combinations of maternal genes and the baby's genes that don't allow these to talk properly, the extravillous trophoblast cells and the NK cells, if you have these, you are more likely to have preeclampsia or a smaller than usual baby or a miscarriage. And this is a really active area of research at the moment, but not one that we can intervene in yet.
So Beth, earlier on, Vicky was talking a bit about what difference it makes if the mother's been vaccinated and had a vaccine recently. Would all the vaccines that the mother's ever had make a difference to that fetus?
Many vaccines would make a difference to the health of the fetus. So Peter talked about some pathogens, some bugs that can cross the placenta. So if we take the example of a virus, which we've got an effective vaccine, is rubella. So rubella, if it caught in pregnancy, can cross the placenta and cause devastating effects in the fetus. It can cause death of the fetus and lots of severe birth defects. But we're really fortunate now that we have a really successful childhood and catch-up vaccination program. So the R in MMR stands for rubella. And because of that, these cases of the fetus catching rubella during pregnancy was hardly ever seen anymore.
And could you then, I was thinking about, Vicky was saying how important it was for, pregnant women to have flu vaccines and COVID vaccines. Could you deliberately give pregnant women loads of vaccines to then protect the fetus and the baby later?
Yes, so Vicky touched on this, so there are vaccines that are primarily aimed at protecting the newborn by transfer of antibodies that then last through the initial weeks and months of life. And then there are vaccines that are primarily aimed at protecting the pregnancy. So for instance, with pandemic flu and COVID-19, we saw large women were at high risk of catching severe disease if they were pregnant and high rates of hospitalization. And these have effects on the fetus. Not only we have maternal deaths, which are very shocking, but also increased rates of preterm birth, stillbirth, and premature birth, which all have long-lasting effects on the health of the fetus.
And Chrissie, what happens to the fetus if a mother catches an infection during pregnancy? We've heard that some things can cross, but what generally happens?
Yeah. So a number of different things can happen. As Vicky said, some of these infections can be more severe in pregnancy, and so we may have a baby who's either born smaller, so small, called small for gestational age, perhaps a miscarriage or a stillbirth. Beth has talked about the case of rubella, where you can get congenital abnormalities, but sometimes you don't get any effects. The baby is absolutely fine. So one example perhaps is a bacterial called group B strep, which many of us will carry in our bowel or in the vagina. So around up to 1 in 3 women in the audience may have this colonizing bacteria, so it lives harmlessly in us. And in pregnancy, that's the same. But in some cases, that can cause infection in the womb and infection of the baby. So for some women, no problems. For others, it might cause stillbirth of the baby, preterm delivery, or the baby may catch that organism as they come through the birth canal and be infected early in life, causing sepsis early or meningitis or sepsis later on. And there are many things we can do to try to prevent these things when we know about them. So we can give antibiotics to the mother for her own health, but also to try to prevent the baby becoming colonized or having severe disease from this. And as we've talked about a number of times, there's vaccines, and we're in a really exciting time. We have 3 different vaccines which are recommended for all pregnant women in the UK, but we're also running clinical trials of vaccines such as that against group B strep. So hopefully in the future we'll be much better to be able to protect these women, not just from colonization, but from severe disease in the mother or the baby as well.
And is it possible then for the fetus to catch an infection while they are in the womb?
Absolutely. So the group B strep might be caught there or as the baby comes through the birth canal. But another example is a virus that many people have never heard of called CMV or cytomegalovirus. And again, in the audience, probably half of you have had this virus at some point in your life and would not know anything about it. And for most of us, it causes us no problems whatsoever. But if you catch this virus when you're pregnant, in a proportion of women, that can be transferred across the baby, and the baby becomes infected as the virus goes across the placenta and infects the baby. So the majority of these babies will be born very well and have no signs and symptoms, but up to 1 in 4 of these babies can have lifelong harms associated with this. So hearing loss or developmental problems in up to 1 in 4 of these babies. So there can be a massive spectrum from a mother not being unwell at all, a baby having no problems, perhaps having some mild changes, or babies who are very, very severely affected. So it is very, very variable.
And Lindsay, we haven't talked yet about what's happening to the mother in the environment. Can the mother's diet or stress levels make a difference to how susceptible the fetus might be to disease?
Yeah, I mean, it's a really important point, because our microbes, I think Vicky said, we're just not, they're not bystanders. They're really fundamentally important for our overall health. That's for maternal health, fetal health, and infant health as well, and actually lifelong health. And actually one of the main jobs that they do is they break down the food that we eat. So of course, mum needs to eat because she needs energy. And of course, the fetus needs energy to grow as well. And actually one of the other key things that happens with the placenta is key nutrients are transported across the placenta from mum to the developing fetus, so it's as strong as it can be. So diet is very important. So kind of a good balanced diet, making sure that there's beneficial compounds that are broken down by special friendly bacteria that are able to go over and help the fetus in that kind of developing process. And the other thing in terms of about the stress side of things, so the other things that we kind of talk about and is kind of much more in the agenda now for the microbiome is the gut-brain axis. So in terms of that, the gut microbes can actually communicate with the brain, and it's a two-way street. So actually there's many evidence before we realise that microbes might be playing a role here. We know that stress is fundamentally important for fetal development and potentially risk of infection in utero, but also kind of shortly after birth and longer term as well. But I guess we're still trying to figure out what that might mean, and if that's purely to do with changes from just stress hormones for mum, or if we've also got microbes playing a role there as well. So that's definitely kind of watch this space.
And that's a hard thing to know though, isn't it? Because it's bad enough being really stressed without then thinking, is this going to have effects?
Exactly. And that's why it's really important to kind of understand how these complex reactions occur. And actually it was a really interesting story. I don't know if some of you remember when Greece had the huge kind of financial crash. One of the things that happened, of course, everyone was very stressed because of in terms of the economic uncertainty in Greece. One of the other things that happened there was preterm rates went through the roof, and they linked that back to potentially stress. So there's lots of other things that we need to consider, not just in kind of, I guess, physiologically, although it is, we actually need to think about the other wider environment as well.
Well, let's take some questions from the audience now on anything to do with the immune system and pregnancy. Please save your questions about after birth and the babies then. But if anyone's got a question, do put your hand up. Yeah, there's somebody right at the back up there. Please do wait for the mic to get to you so that we can hear it.
Hi, thank you for the really interesting discussion so far. So thinking about really the mother for a bit rather than the child, obviously the immune system is going through huge changes during pregnancy. How long lasting are these changes? Does our immune system completely recover to pre-pregnancy state after, or do we keep some kind of tolerance, especially to the fetal antigens, even after 2 years, 3 years after the pregnancy?
Who'd like to take that? Vicky, that sounds like your area.
So I think there are kind of two parts to this question. The first is how quickly does the immune system change back? And the second is, do we sort of maintain tolerance to really the placenta? And one thing that I've always found absolutely fascinating, and I think it's not well understood at all, is I mentioned that these extravillous trophoblast cells, they invade into the lining of the uterus that actually mixed in with the mother's own tissue. And when birth happens, most of the placenta comes out, but these cells will stay there for a couple of days, a week, but they ultimately will get kicked out shortly after birth. What is it that changes that allows that to happen? I don't know. The other question is, does this slightly different immune response continue after birth? And birth is a massive challenge for the immune system. Birth is probably largely probably at least partly mediated by the immune system. I'm biased, obviously. But thinking about the immediate postpartum period, we do see things start to go back quite quickly. But one of the things that makes a big difference is whether or not people are breastfeeding. And I think the best evidence of this comes from studies of vaccine responses, looking at people who aren't pregnant, people who are pregnant, people who are breastfeeding. And actually, if you're breastfeeding, it does look a bit more like the response when you're pregnant. So I think you kind of maintain a little bit of immunological pregnancy-ish in breastfeeding. So I think it's more to do with whether you continue to have these different hormones around reproduction rather than the actual amount of time that you wait, if that makes sense.
Is that a good thing that it stays a bit different and a bit like pregnancy, or not a good thing?
I think it's probably neither a good thing nor a bad thing. I mean, I didn't really answer this when you asked me earlier about the immune system being different in pregnancy. Is that actually necessary to support the pregnancy? And we have no evidence from humans, really, that it is, and we have some evidence from mice that it isn't. So my view is that these immune changes that happen during pregnancy, they're more a side effect of things that are going on with hormones, and many sex hormones sort of crosstalk with the immune system. And I suspect it's the same with breastfeeding, that we're just having a hormonal effect on the immune system. But I think we probably couldn't firmly say that based on the evidence at this point. That's just my vibe.
Thank you for that question. Yes, anybody else got a question? Yes, we've got somebody. Oh, yes, there.
Wonderful talk so far. I was interested in, you were speaking about cytomegalovirus and how in about 50% of the population you've been infected and it's harmless. Couldn't you potentially test pregnant women to see if they've been exposed, and if they haven't, is there a vaccine in development that could then give them immunity and protect them and the fetus?
An excellent question. So just to repeat some of the questions, so about 50% of us have had CMV before. And when you've had CMV before, you have antibodies to that virus. And so it makes it less likely that the virus, if that a virus gets reactivated or you get a new strain of the virus, there's this much smaller chance of the virus being transmitted from the mother to the baby. So it happens in around 1 to 2% of the time if you catch an infection for the second time of CMV in pregnancy. If you catch it for the first time, that's more like 30%. So immunity is important in terms of antibody is important in trying to prevent that transplacental transfer. So the next part of the question really is around whether we can test a vaccine which would prevent transmission or stop women getting it in the first instance. And indeed, there are, for many decades, we've been trying to develop a CMV vaccine. But again, in a really exciting time where Moderna have an mRNA vaccine, which is being tested globally, but we're taking part in the UK, and I'm the lead for that in the UK. And we're testing it in non-pregnant women at the moment, and also in adolescent girls as well. The question is when you're going to give that vaccine to the mother, because infection, which occurs very, very early in pregnancy, is that's associated with the most severe outcomes. So if you wait until pregnancy to give a vaccine against CMV, it's basically too late. So you need to be giving a vaccine prior to pregnancy. So then the question comes, do you give it to adolescent girls, but can you get immunity lasting from adolescence all the way through to pregnancy to try to keep high levels of antibody to see this difference in the transplacental transfer of the virus? Or another way that we perhaps will be doing this is giving it to toddlers. So if we give it to toddlers, we can try to prevent them getting it for the first time. And toddlers are the main source of infection to pregnant women around them because you excrete the virus in your saliva and in your urine. So perhaps we could vaccinate toddlers to try to solve this problem in the future. But hopefully a solution is coming, but we're still a few years away.
That's really interesting. Thank you. Yes, anybody else got a question? Yes, there's two questions just here. We'll have the one near the front first.
Thank you. I just wanted to ask that I've read a few articles talking about fetal cells being present in different areas of the mother's body years and even decades after delivery. Why does that happen, and why does it does it affect the baby's immune system into adulthood or the mother's immune system after pregnancy?
Vicky.
Yeah, really good question, which I'm going to partially answer the mother's side, and I think I might throw it over to Petter for the baby. So yeah, there's evidence that this can happen, and there is also evidence that if any fetal cells end up, kind of lodged, I guess, in the mother, this can change immune responses. And in fact, not in necessarily a good way, because there has been an idea, I don't know if it's necessarily continued to hold up, that some autoimmune diseases, like for example, scleroderma, which mainly affect women and mainly affect women who've had children, these might be basically a maternal response, not actually to her own cells, but to these cells that are left lodged in the body after pregnancy.
That's fascinating. And Petter, what about the
Yeah,
baby?
this is a great question and a very interesting topic. As you would imagine, it's incredibly difficult to study this in humans, right? Because you have individual small numbers of cells anywhere in your body, and you're not going to find them in a small child, right? So most of our data comes from mouse models, so mice. And there's evidence that tolerance can develop towards the mother's cells. And that's why they are still around for many, many months in the mouse. But whether this is any relevance or has any relevance to human disease or function of the child's immune system is too early to tell, I would say.
Thank you for that. And yes, there was somebody a couple of rows behind. Yes.
When you showed the picture on how all the blood vessels changing, it made me think a little bit about how the immune system is structured within the uterus. In the gut or in the skin, you have certain centers of immune cells. So I was wondering, how is that in the uterus and how is it changing during pregnancy? Thank you.
That's a really good question, and it's one that we're only just beginning to properly answer, because for many years we've sort of had to go, but we've only been able to look at one thing at a time. And with new technologies, we can now produce just the most beautiful images showing everything that's going on. And I'll give you some highlights. We see the NK cells interacting with the extravillous trophoblast cells. We also see macrophages interacting with them. That's almost certainly important as well. And then we also see things like aggregates of immune cells, things like T cells and even some B cells, deeper down into the uterus. And then we also see some quite up near the top, and they're probably to do with defense against potential infections. So that's a kind of bird's eye view of how they're located in the uterus. And it gives us a lot of clues as to what they're doing if we think about what cells they're talking with. And trying to work this out, trying to map it in a lot of detail is something that's been a big concern of research in this area for the last 5 or so years. And it's somewhere where we're making a lot of progress, but I would still say that we're not completely there yet.
Well, we'll take some more questions again later on, but we're going to jump now to the end of pregnancy and to the birth and afterwards. So, Lindsay, if I can start with you, how is the newborn baby's immune system affected by the type of birth, whether there was a C-section or whether it was a vaginal birth?
Hot topic. Fantastic question. So, and I think probably one that everyone wants to kind of know the answer to. So this is really, really important because, as Vicky alluded to, the fetus is sterile, so there's no friendly bacteria there. Potentially there's bacterial kind of compounds or small molecules that cross the placenta that get in, but not whole bacteria. Or if they do, it's normally a bad thing for the fetus in terms of infection and potentially complications. So in terms of how do they get these first friendly bacteria, this happens at the process of birth. So actually what we normally find is certain types of friendly bacteria are vertically transmitted from mum to baby during vaginal natural childbirth. So it's a perfect way to seed the baby, to give the friendly guys immediately over to the new baby to kind of help them get the immune system up and running. So if you think about it, that's kind of what happens naturally, because these microbes are passed on actually from the vagina, but also actually from mum's gut is also a really kind of rich source of these friendly bacteria. So if a baby's born by caesarean section, you basically just bypass. It's the ring road. You're not going through town, you're going round town. And in this case, that's a bad thing, because you then miss out on potentially getting these beneficial bacteria that way. And it normally means that caesarean section babies have different types of microbes. They might have microbes found on the surface of the hospital, skin microbes, for example. And of course, these microbes are really important for programming your immune system. So if we don't get the right ones in during caesarean section, it normally means the immune system isn't programmed the same way as you getting these beneficial bacteria through this through this natural childbirth. And actually, there's lots of epidemiological data showing that if you're caesarean section born you've a much higher risk of developing allergic diseases, kind of short-term and long-term, and also autoimmune diseases and chronic diseases as well. And we're now beginning to realise it's probably because the bugs, the correct bacteria, the correct viruses, the correct fungi are not there to program the immune system in the best possible way.
And so one of the solutions that's been suggested to this is to deliberately, if a baby's been born by C-section, for them to then take a vaginal swab and wipe it on the baby's head so that you get the bacteria there, which sounds like such a good idea, and then but it doesn't get done, does it? And lots of experts I know I've talked to say, oh no, but this would be intervening and might not be a good idea.
And that's controversial. So we've gone from hot topic to controversial. So, and actually I'll probably let some of the other panelists introduce this, particularly the clinicians. So I think we have to be really careful here. I mentioned about some of the bacteria being passed on potentially kind of from natural birth through the maternal like vagina, but mum's gut bacteria are really, really important. So a vaginal swab is not going to transfer those friendly bacteria over. So for example, bifidobacterium, which is one of our favourite bacteria that we work with, that's really important in early life, and it's vertically transmitted from mum to baby during natural childbirth, and it's basically the main source of it is mum's gut. And actually the other thing that's really important here, which we've already had the introduction to, is group B strep, which potentially in the fetus and neonates is potentially deadly. And that's something that could potentially be passed on by doing this vaginal swabbing. So you have to be really careful, and this is why we need more research in this area. But this is not a case of like, if you do a vaginal swab, it's going to sort absolutely everything. It's much more complicated. And also from my perspective, I would much rather put in the back-end bacteria, we know exactly what we're putting back, right? So I think that's really, really important. Probably Petter can answer that as well.
Yeah, so I just want to moderate the view about cesarean section versus vaginal delivery a little bit. You know, oftentimes cesarean section is done after the membranes have ruptured and the baby is actually exposed to the birth canal, and then the delivery has to be transferred into a cesarean delivery for emergency purposes. That is likely not as severe in terms of the abnormal seeding of microbes as the planned cesarean, which is done earlier than expected due to other reasons. So that's one thing to keep in mind. The other thing is that oftentimes the babies are exposed to antibiotics as a consequence of the cesarean section, which also has important impacts. And if we sort of take away those effects, the actual effect of the C-section itself is not that great and not that important.
And Chrissie, you wanted to come in on this?
Yeah, so I've had 2 C-sections, both emergency, both not planned. But just in terms of if other people are worried about this and, you know, what the effects of their babies might be, there are other ways as well that we can colonize the babies with healthy bacteria or friendly bacteria. So skin-to-skin contact shortly after the baby's born, breastfeeding, these are all really important interventions to make sure that the baby is being colonized with those organisms from the mother. We've also been trying to manipulate this a little bit as well by giving friendly bacteria deliberately to the mother to see whether we can cause more of those friendly bacteria to go across to the baby, so colonising the mum's nose and seeing whether we can manipulate the system a little bit that way. So there are ways to do it, but as Lindsay said, we've just got to be so careful that we're doing something that's safe, because in uncontrolled ways, we just don't know what we're introducing. And there are many bacteria that live in the gut and the vagina which cause severe disease in babies. So it's a case of wanting to do good and not wanting to do harm.
And what about, Chrissie, if a baby's born prematurely, how does that affect their immune system and their ability to fight infection?
Yeah, so we've talked a little bit about how the immune system's adapted in the mother, in the fetus, and also in the newborn baby as well. And those differences are evident in term babies compared to adults, but they're even more pronounced in the preterm babies. So there are a number of different changes which are evident in a preterm baby compared to a term baby or an adult. And that includes both on a cellular basis, so a type of the white blood cells called the neutrophils, which help protect against particularly bacterial infection. They're both lower in number, they're slower to move to the area where they're needed, and they also function less well. So we do know that rates of severe and invasive disease are higher in babies, and this may be one of the contributing factors to this. Vicky so elegantly described to us how antibodies transferred across the placenta from the mother to the baby, and a lot of that is occurring towards the end of pregnancy. So if a baby's been born prior to the time at which most of that antibody is going across, they're not benefiting from that antibody. And the second part of the question was the consequences for infection. So we do see higher rates of infection and higher rates of infections that may have come from the mother. So for instance, the herpes simplex virus, which causes cold sores and genital herpes, we do see many more, an overrepresentation of preterm babies compared to term babies, probably because they've not had so much of the antibody coming across the placenta to the baby. And also for conditions which we can vaccinate against, such as whooping cough or pertussis. The mother may have had the vaccine well before the baby's born, but there hasn't been enough time for that antibody to go across the placenta to the babies. And preterm babies have about 4 times higher risk of severe pertussis compared to term babies. So yeah, really important. And we are much more vigilant about these babies. On the neonatal unit, we intervene much more quickly because we know they're much more vulnerable to severe infections.
Can I follow up on that real quickly? So another reason why they're more susceptible to infections is also that their barriers are imperfect. You know, the skin is thinner, it's not as protective, the intestinal barriers similarly. So I think that's another point of view to keep in mind, that it's not only about the function of their immune cells, it's more about the conditions in which they come out. They're not ready to come out and meet the world outside. So I think that's one thing. And the other thing is we put them in intensive care units where we put needles in them to give them blood and fluids and things. These things, even in an adult, increases your risk of severe infections. So that's true also for the preterms.
And Beth, why, when you've got babies born at term, and of course preterm babies as well, but why at the right age is are vaccinations so important for the babies when it comes to their immune system?
So we've heard a lot about how antibodies transferred across the placenta, and this provides this passive immunity, we call it. So it's just the transfer of antibodies, and these decrease with time. They also receive some antibody through breast milk, but at some point the baby has to produce its own active immune response to bugs, to viruses and bacteria, and that could either be through them encountering an infection, which would make them sick, or be through vaccination. So what we really want to do is have deliver a vaccine, which means they can mount their own active immune response and give them memory to respond to that infection in later life. And we want that to do that as early as possible before they have the chance to be exposed.
Yeah, so that they're not getting ill. Yeah. And Vicky, how exactly is maternal milk helping the immune system? What do we know about, for those who can breastfeed, what do we know about what is working there?
Less than we should, I think. But yeah, I do want to start by saying that breastfeeding can be really tough. I certainly found it really tough. And so even though I'm going to tell you about some of the benefits of it, if it's really not working for you and your baby, I don't think that in a country where we have clean water, these benefits are so much that you should beat yourself up if you can't do it. But what is in breast milk? Well, we do have some evidence that it reduces the number of the squirts in babies and how long that goes on for if the babies get sick. Also, it seems to reduce the number of ear infections. And we also have a really interesting study that looked at giving rotavirus vaccine to babies, which is a live viral vaccine, weakened, and babies who were breastfed actually shed less of that into their nappies than babies who are not breastfed. So that's kind of like a model of how breastfeeding is reducing the growth of a virus inside a baby's gut. How's it happening? Well, there are 2 ways, really. There are kind of general antimicrobial components in breast milk. So there are things called glycoproteins, which are kind of basically sugary proteins. And these can act as sort of like a non-stick coating on the gut of the baby, which prevents certain bad bacteria from getting a foothold. There's also something in milk called lactoferrin. Milk is full of iron because babies really need iron, but bacteria also really like iron. And so lactoferrin kind of binds onto the iron and grabs hold of it, so the bacteria can't get it. It's only released when it's ready to be absorbed by the baby. But then we also have antibodies. So those are kind of general things that are just blanket suppressing particular bacteria or viruses. But antibodies in breast milk, those can also protect babies against infection. And to me, the best evidence of this is there was a trial called the Mother's Gift Trial. This was the trial that showed us that flu vaccination in pregnancy reduces the chance that babies will get flu-like illnesses when they're after birth. But what was really interesting in this trial was they also looked at the people who got vaccinated but didn't breastfeed compared to the people who got vaccinated but did breastfeed. Did the breastfeeding provide extra protection against the flu? And it did. And it's not exactly known how it's working, but we do know that breast milk, if you mix it with, say, flu virus, or also if you mix it with COVID, and then you try to infect cells with those viruses, it's less easy to infect the cells with those viruses. So probably something similar is going on in the baby's gut.
And do we know, Lindsay, what the ideal microbiome would look like for a baby? What would it be ideally in the baby's gut? Because the moment they get to their new home, they're starting to get colonised by everything, aren't they?
Yeah, and actually there's some really cool work showing that, unsurprisingly, maybe if you've got pets at home, the pets and the babies normally share quite a few microbes together. Which again, because your microbes are everywhere, so you pick them up everywhere. To go back to the breastfeeding point as well, the other thing in terms of how that can also influence kind of immunity and thinking about what your perfect microbiota potentially looks like. Breast milk also has really specific dietary components of very special sugars called human milk oligosaccharides. They can act as decoys, as Vicky's already introduced. But the other thing that's wild about them is that they're relatively complex, and the baby themselves cannot break these specific sugars down in their gut. It's the baby's friendly bacteria that does that job for the baby and then releases the special compounds to either program the immune system to strengthen the barrier and to do a whole host of other things. So it's absolutely wild that we've evolved that there's something in our milk that doesn't feed directly the baby, but feeds the baby's friendly bacteria, who then feed the baby, which is kind of like amazing. So in terms of, I guess, what does a perfect microbiota look like, that's really tricky because we need to kind of go in and find out what's there under normal, healthy conditions. And the other thing that we've done a lot of studies on this, but most of this work has been done in high income countries, so kind of Europe and North America. And we don't really have a good handle on actually what these bacteria looks like right across global infant populations. And that's something that's really important to do because we know there's geographical differences as well. But one thing I think that would be characteristic would be normally a beneficial kind of microbiota would be lots of bifidobacterium normally, because bifidobacterium's favourite food is milk sugars. So they're perfectly evolved together. And Petter will probably want to follow up on that for all the fantastic work they've done as well.
Yeah, one of my favorite topics. So one thing that's really interesting about these specific bifidobacteria, for example, there's a particular strain called Bifidobacterium infantis, because it was found only in the stool samples from newborn babies. And what's interesting about this bug is it seems like a super bug for young babies because it really trains your immune system to dampen inflammation and promote health. The problem is that in this part of the world, in Europe, in America, in industrialized countries, this bug is more or less gone. About 4% of babies naturally have this bug. But if you look at samples in Bangladesh, where in sub-Saharan African populations, 95, 100% of the babies have this bug, which is probably something that we've evolved with, that is good for us, that we have depleted through rounds of antibiotics in mothers for generations in this part of the world.
That's absolutely fascinating. And you mentioned, Vicky, that there can be long-term impacts, really long-term impacts, if a baby has an infection early in life, long-term impacts such as being more likely to develop asthma. Do we know why there could be these long-term impacts?
Yeah, so this is a topic that's close to my heart because my little girl as a toddler got RSV, which turned into viral wheeze, which is now asthma that she still has. And this is a good example of an infection that we think continues to affect children later in life. But this was first observed in the 90s and has continued to be since. And if you get RSV as an infant, you're about 2 to 4 times more likely to go on to have asthma, depending on the study. But it's really tricky because we can't really tell the difference in a whole messy human population between RSV causes asthma or increases the risk of asthma, and babies who are more likely to have RSV are also more likely to have asthma. Now, there is some evidence from mice, if you give them RSV, that certain immune molecules that are associated with asthma, one of them is called IL-33, get upregulated in the lungs. So that gives you a hint that maybe it is causative. But one thing that I think is really exciting, that we've actually already touched on with Chrissie, is we are developing new vaccines that are designed to be given in pregnancy in order to protect babies in early life. And one of these new vaccines is the RSV vaccine that they've started giving in America this past winter. We're not giving it here yet. And hopefully this works really well because RSV is grim. It actually causes more than 50% of hospitalizations and deaths in babies under 6 months old. And what I would be really interested to see is if we start using this vaccine and it successfully reduces the rate of RSV, will we see a reduction in the rate of asthma? And I think that's something that's really interesting to keep an eye out for.
And Petter, are there any other ways in which exposure to infections very early in life can have long-term
Absolutely.
consequences?
I mean, the antibiotic use is a really good example that Vicky already touched upon, which is that some babies have to get antibiotics because an infection could be lethal early in life. But it also sort of perturbs this natural process of colonization by friendly bacteria, which in turn has long-term consequences for health. And we think that the way in which this happens is because the immune cells that are developing and sort of getting to know their microbes, allowing some to stay and others should be rejected. If you perturb that process, you're probably sort of triggering a negative vicious cycle, which when you get infected could lead to more severe immune responses, causing inflammation, tissue damage, etc. And so we think this sort of, there's a critical window during the first few months of life when your friendly bacteria needs to be permitted, to settle down and establish. And if you perturb that process, it can have long-term consequences.
That is going to be really hard to avoid, though, isn't it? When, as you say, babies can get sick very quickly, and some will desperately need antibiotics.
We know that breastfeeding is one thing that mitigates this negative effects, but we need to develop strategies to counteract some of these negative effects. Yeah. And that's what we're working on.
Well, I'd like to take more questions now. So has anybody else got a question they would like to ask? Yeah, Zoltan's got our online questions for us. What do you have for us?
A couple. So yeah, I'm fielding questions from the audience online. So Vicky, in your talk, you mentioned that placental villus cells lack MHC. Is there any evidence for viruses which exploit that? I'm thinking maybe Zika or something along those lines.
Ooh, that's a really good question. I don't actually know if I can answer it. Can you answer it? I mean, one would think from how the MHC works, and I can tell that this is a very educated question that's come in.
That's what I thought.
We would think, based on how the MHC works, that this might make the placenta more vulnerable to viral infection, and yet most viruses can't infect the placenta because, in fact, the placenta has lots and lots of we would call them restriction factors, but basically innate antiviral factors that stop viruses from growing in it. So it's a great question, but I'm not sure that the MHC is the most important thing for placental cells.
Okay, thanks. One more, and I'm guessing this will be a question for Lindsay. Is there any evidence for taking probiotics during pregnancy? It's always going to be a probiotic question, right? Is beneficial for both the mother's immune system and protecting the fetus and the fetus itself?
So it's a great question, and obviously one that everyone's kind of wants to know, and kind of going to Petter's point about, coming up with new therapies, maybe microbial or diet derived. So I guess there has been some work showing that probiotic supplementation during pregnancy, but then also following up that probiotic supplementation in early life does appear to reduce atopy, so like kind of eczema, but they haven't found it for asthma. I don't think it was protective against that. But again, most of these trials have been relatively small in number, and they maybe haven't been designed in the most optimal way. So I guess the jury's still probably out, and this is why we need to do these interventions, but Petter will be better probably.
No, you're absolutely right. And one of the problems with the probiotic, it's a large industry that, promotes this type of work. The problem is that we are not evolutionarily designed to have a monoculture, meaning everything is just one bacteria. But what happens if you just keep giving a tablet every day with one bacteria is you're basically going to have that take over your colony, if you will. And that's not really the way we're designed. And so, cautious.
Yeah. Thank you. Yes, who else has a question for us? Here. Yes, one just here. Thank you.
And really just to follow up on that, how do we know how the microbiome of the mother while she's pregnant affects the fetal immune system, and what might be the theoretical or what experimental evidence we might already have about how that might act?
So it's really interesting because actually if you just profile the microbiome over pregnancy in mum, it changes quite significantly. And potentially this is also linked to all of the sex hormones that Vicky's talked about. There's kind of all these changes as well. So these microbes are actually responding, which kind of makes sense. Because one of the other things that happens actually that mums, for example, bifidobacterium levels go up kind of during the course of pregnancy, which kind of makes sense because then mum's prepped and ready to pass on her friendly bacteria to her baby. So that kind of makes sense. Most of the work, again, would be, it's very difficult to do these studies with humans. So most of the work has been done kind of with mouse models, preclinical models. So, and it's well known now that potentially these microbial metabolites can cross over the placenta. We've even done a little bit of work of this ourselves, showing that BIF-derived kind of small compounds, bifidobacterium compounds, are actually helpful for placental kind of development, and also actually programming kind of the fetal liver and the intestine with some kind of immune things in there as well. But I guess we've looked much more at the microbiome after birth, and I think we've got a lot more to do on that side of things, but I don't know if Chrissie or Beth or, well, everyone actually has something to say about that.
I mean, there are just these huge amounts more to understand about the microbiome and how that interacts with the immune system, how that interacts with health. There certainly are different patterns of the microbiota which predict long-term health in infants. But again, just understanding those distinct patterns and exactly how they affect long-term health. And also coming back to the previous question about probiotics, is how can we effectively manipulate that? And probiotics are challenging because probiotics aren't one thing. They're uncontrolled amounts. They're different things in different preparations. And I think what we need is better experimental models so that we can more accurately define what's going in, so exactly how much is going in and exactly what's going in with human challenge models, to be able to understand what the longer term consequences are and how we might manipulate health and the immunological system associated with that. So I think the way to go is experimental human challenge models.
Yes, somebody just here.
Hi, I have one question about the composition of breast milk. So I don't know if it's actually myth or fact that if a baby is ill, or actually at the beginning of being ill, that there's a feedback to the mother that more antibodies are secreted into the breast milk, and so you push the passive immunity to the baby. So if somebody can comment on that, how does it work?
I can semi-comment on it because I've seen the evidence for it and it's quite weak. So I wouldn't want to bet either way myself personally, but we do see the composition of breast milk changing in response to illness, and particularly things will change in terms of the amount of fluid that goes into it. You quite often see more fluid in breast milk in illness.
That's amazing. Thank you for that. Anybody else? Oh yes, there's one over there near the back.
Thank you. So you mentioned how it's the sex hormones that might be modulating both the immune response and the microbiome. Do we know how hormonal contraceptives might be affecting immunity and the microbiome in non-pregnant women?
Contraception and the microbiome?
Great question, my next research grant.
It is a fantastic question. Actually, I've got a colleague over in Germany that's kind of actually looking at this as well, because of course menopause is another time when all your hormones change as well. So actually we do know that, again, the composition of the microbiome will potentially change, but there's always kind of a feedback loop as well. So, but again, we need to do those studies, and we can't just do them at one point in time. We need to kind of follow up as well. And of course, doing that in humans is a wee bit challenging, depending on when you're going to look at, but it's a really important question as well. I mean, obviously, in terms of, yeah, so I think that's watch this space, but as you say, yeah, there we go. We've all got a joint grant that we can put in, so thank you very much.
Yes, another question at the back there.
It's really interesting, thank you. Perhaps I've misunderstood something, but how is baby inoculated with the maternal gut microbiome, or is that just something that happens passively over time?
They're really close.
Yeah, that's the answer, yeah.
So during childbirth, basically, there will be
delivery.
Crossover during delivery.
And those of you who've had a baby know that there can be quite a lot of crossover, can't there?
Hello. Just keeping with the theme of the composition of the microbiome in the mother, how does the maternal age affect that, or does it actually affect what is passed on to the baby, the microbiomes that the mum has?
That's a really interesting question. And actually we know, so actually it's normally you kind of get your adult-like, I guess, microbial community by the time you're about 3 or 4 years of age, which is pretty young given by the time we think that we would be adult or like childbearing age. But we do know at older life stages that the microbiome composition changes again. But again, I don't think we've got those studies. We haven't been able to power them enough, have enough women in these different age groups to have a look. And I would say as well, age is probably important. I would argue that probably lots of other factors like antibiotic usage, diet would have a much more overarching role in terms of microbiota composition and what that might mean for mum and also for the fetus and for the baby as well.
Thank you. Yes, there's a question there.
So I'd like to ask a little bit more about going to autoimmunity. So it's been stated that mothers their immune system shifts during the pregnancy. And I was wondering, for mothers that already have a substantial shift because they are autoimmune, such as those who have multiple sclerosis or lupus, what additional or even diverse changes happen to their immune system during the pregnancy, which are otherwise not usually things that mothers need to worry about?
Petter.
I think I understand what you're saying. That so think of it this way: certain branches of your immune system are detrimental for implantation and pregnancy and fetal development. Those need to be tuned down in the mother to allow this to happen, as Vicky illustrated, right? Then other things can be boosted to provide that protection from viruses, for example. And one of the things that can be boosted is something called type 1 interferons. They interfere with viral replication. They drive diseases such as lupus. And that could explain why, for example, lupus gets worse during pregnancy, but that thing that is dampened might drive another type of autoimmune disease, and thereby explain why that particular disease gets milder, typically, in pregnancy. So it's all about tuning different branches of your immune system up or down.
Thank you. Any other questions do we have? Oh yes, sorry, one right up the back there.
Thank you so much for the talk. It's less of a scientific question, I guess, but being experts in this area, how do you feel like personally, with so much knowledge in pregnancy and in childbirth, how does it affect the way you look at your pregnancy? Because some of you mentioned that you're mothers. Right, yeah. Some of you mentioned that you're mothers, and does that influence some of your actions? And how do you, balance kind of the scientifically, I know maybe this is the better way of doing it, but like I want to do it the other way, maybe.
That is such a good question when you do know so much. So you mentioned you've had children in C-sections. Did you think a lot about the science of it when you were pregnant?
I was a total nightmare being a pregnant woman. So I'm a paediatrician, and when you're training, you go to only abnormal deliveries because no one needs a paediatrician when everything's going smoothly. So I did not believe a normal delivery was possible, and clearly it was not possible in my case. I'm a clinician, so I'm probably quite a pragmatist as well. And I do have a trust in the healthcare system and the guidelines which are in place. And there is a lot of thought that goes into those things. So although all these questions are fascinating and scientifically enriching, we should definitely be pursuing all these things. I think in my own pregnancies and perhaps with my children growing up, I do have a trust that we have developed guidelines. We've put a lot of thought into the interventions for pregnancy and in childhood. And so I think trusting that you are in good hands with your clinicians looking after you, that the vaccines which are recommended in pregnancy now have a whole host of evidence to show they're safe and effective, that you don't necessarily need to know all of that data, but you know that the NHS has put a lot of thought into what's recommended. And so I think it's a balance of, for some people there's a high need for information, others it's recommended and so you'll get it. So I hope that partly answers, but Beth, you've had...
I can add something. I've got a 2-year-old son, so I would say I actively sought out the vaccines during pregnancy. So if they weren't brought up in my appointments, I asked about them, and so I was quite proactive at getting whooping cough vaccine, flu vaccine, and also COVID-19 vaccine, which at the time was very early. So I had my first dose before I was pregnant, and then my second dose during pregnancy. And actually it was interesting because when I was waiting and looking at the consent forms, they actually said you had to indicate if you were pregnant, and it was almost like it was an exclusion criteria. But I knew the information, and I ended up having a conversation and telling my vaccinator about why it would be safe in pregnancy. And the other thing I did...
So you were a nightmare pregnant woman too.
The other thing I did, as I went for a private Group B, I did a private Group B strep test as well, to find out my Group B strep status, which I did by post.
Yeah. So you were being influenced by knowing there's so much about the science. Yes. Yeah. Yeah. And Vicky, because you mentioned, of course, your child having RSV, and you must have known ever such a lot about that.
No, I didn't. I really actually didn't know very much about it at all until it affected me personally. So I think this tells you, that actually the personal knowledge is quite important. But I guess for me, I really agree with what Chrissie says. I, in both of my pregnancies, just kind of went to a place where I let it happen, and I took my midwife's advice, and I loved my midwife. And the only thing I've probably done that's different from the NHS advice is that chickenpox vaccine, I believe, is now going to be offered generally. But at the time that I had both of my children, it wasn't. So I went out and got it at a travel vaccination clinic. And I was super glad that I did, because each of my kids have been the only child standing in a chickenpox outbreak that's taken down their entire class.
Very good. Anyone got any final questions? Yes, there's one there.
Thank you so much. I really enjoyed the session. I'm convinced in my pregnancies that my pregnancy with my son was way more difficult than my daughter. And I was just wondering if there's any evidence that sex of the fetus affects the maternal immunity, whether it's regulatory or pro-inflammatory, and does this have an effect on the mother long-term in their health?
Any evidence on that?
I can't really comment. I can't really answer your question fully, but I can say there are lots of sex differences in the placenta, which is super interesting and something that we're studying. So male fetuses are at higher risk of developing severe infection, and if there's chorioamnionitis, which is infection of the placenta and the membranes, that's more severe if the fetus is male. And we're studying at the moment how the placenta is different between male and female fetuses, but I don't know about the maternal aspect.
Well, before we wrap up, I want to ask each of you on the panel what you think the future looks like for our understanding of early life immunity and improving long-term health impacts as well. Beth, let me start with you.
Oh gosh, okay. So the future...
It's good to go first, otherwise everyone else says what you're going to say.
That's true. Well, I'm not sure if anyone will say this. So I would say something that's close to my heart is developing good in vitro models of the placenta. So we have a project at the moment which is using primary placental cells isolated from term placenta, and we're trying to develop we call it placenta on a chip in the lab. And we're hoping that this will enable us to study exactly how antibodies cross the placenta, and importantly study like novel antibody therapies which we can't sort of test immediately in humans. And that's really important because there aren't really there are some good animal models of pregnancy, but most of those are non-human primates, which are very expensive and have ethical constraints, whereas mice have quite different placentas. So we're hoping that might be the future.
And Chrissie, where do you think things are going, or where do you want them to go?
I think we live in an extremely exciting time, actually, with pregnancy and early life. And Peter and many others have contributed a huge amount to our understanding of early life immunity and pregnancy immunology as well. But we live in a time where we have 3 vaccines which are recommended in pregnancy, and we have 2 more against RSV, against Group B strep, which are in late phase clinical trials, which would make an absolutely enormous difference if we could protect babies against RSV. As Vicky said, a major cause of hospital admission. In the hospital over the winter, our wards are packed with children with RSV. Again, protecting against group B strep. Those are the most severe infections that we see in small babies, causing sepsis and meningitis. If we could take that out, I'd be laughing, I'd be on holiday a lot more. So I think those are major things. CMV vaccines, which are not in pregnancy but pre-pregnancy, again would potentially hugely reduce the burden of hearing loss and neurodevelopmental problems in children. So I think really exciting times ahead. With lots of hope for trying to reduce morbidity and mortality in pregnancy and in early life.
That's good. That's nice and hopeful. Petter?
Yeah, I share that enthusiasm about the vaccine development. Yeah. For me, another thing it's really an epidemic is all of the allergies and immune-mediated diseases that are just on the rise in this part of the world, but now increasingly all across the globe. And there's a lot of evidence pointing towards something going wrong during the first few months of life when these microbes colonize the baby and the immune system of the baby is evolving. And so we are really excited about now, for the first time really, being able to study this directly in human newborns, comparing kids from different parts of the world where the rates of these diseases are very different, and then learning the mechanisms of how this thing actually happens so that we can give every child a better start in life, basically, and hopefully reduce some of these rates of many different diseases.
And Lindsay, what are you looking forward to in the future? Where are we going?
A little bit of everything. So in terms of the development stuff, I think Beth's saying about the new kind of like in vitro systems, the placenta on a chip, that's fundamentally important. If we are going to develop a new microbial therapy, we need to test, we need to figure out how it works. We need those wonderful systems to be able to do it. And then we also need to understand how these microbes potentially also boost vaccine responses as well. So that's another way that these microbes can work. And just kind of to reiterate, we need to know what are those beneficial bacteria and exactly how they work. So we need to get down to that, we potentially might give a drug. We know how that modulates, what that does in the body. That's what we need to get down to the level of these microbes. Unfortunately, it's a bit more complicated because there's quite a lot of these microbes. So there's a lot of work to do. But I guess that's kind of I think we're making fantastic strides on that moving forward. There's some really exciting big projects happening globally, and there's lots of efforts on it. So it's definitely watch this space, but I think it has already taken off, and I think will continue to fly.
Thank you. And Vicky?
Always a challenge to go last. Everyone else has said such amazing things, but I guess I would say that all of these advances, new vaccines potentially modulating the microbiota, developments in research that will allow us to understand more about how antibodies cross the placenta. These can make massive differences to human health, but only if people feel comfortable using them. So I think a really big challenge, and one that we've kind of started on tonight, so thanks all for being part of it, is making sure that people understand what we're doing, why we're doing it, what we've done to make sure that things are safe, how much we care about advancing human health. And you've all been part of that tonight, so thanks very much.
Thank you very much.